Welcome, my dear students, medical students, aspiring doctors, fellows, and the cardiologists the world over. This is Dr. Jaitly. I will be talking to you in this video presentation a very unique set of things that are present within the myocardial cell, and that's called the sarcomere. Now that's the contractile and the relaxation unit situated within any cardiomyocyte. By the way, this video is a sequelae of the video number 112 that is also on cardiomyocyte. So it's a follow-up on that, just to let you know. On the previous one, just to recap quickly, I discussed what a myofiber is, which are striated heart muscle visible to the naked eye. But then when you look at under light microscopy, you will see tons and tons of these myocytes, which are nucleated, obviously, and that's what makes the striations seen on the myofibers. Each of these myocytes, they have their own organelles, just like any other cell, but obviously the cardiac cell is uniquely situated to perform its function called contraction and relaxation, and that's what we're going to delve in this video very shortly. So the myocytes, they obviously contain the various organelles like Sarcoplasmic Reticulum. This is the sarcoplasma, which is the outside membrane, which invaginates within. There's a nucleus which contains the genetic information, obviously, that determines what kind of a cell, or the longevity of the individual perhaps will also be determined by the genetic foundation, which is situated within the cell here. And then you have mitochondria situated within the cell here, which are the ATP stores, if you will. And it's like your gas tank of your car, and essentially stores all the fuel and the energy for the cardiomyocyte to contract and relax. However, each of these cardiomyocytes, if you were to look under electron microscopically, you will see a unit called sarcomere. And that's what we're going to discuss in this slide very shortly. So the sarcomeres now, as we know, they are situated between the two Z lines. Now, they are the contractile proteins which are uniquely present within the cardiomyocyte. And there are tons and tons of such numerous sarcomeres within the cardiomyocytes which constitute these filaments. Now, the filaments obviously are within the myofibrils, and there are three types of filaments, if you will. One is an actin, the other is a myosin, and third is a titin, which is only recently discovered, like I said in the previous video. Actins are very thin filaments. Myosins are the thick filaments. Now, the red one is thick, as you see, that's the myosin. The blue ones are the actins. I've tried to sort of color code and match them here for you. And these are the Z lines, which are in the green. Now, remember, this is all electron microscopically we are looking at within the cell. So this is the layout of the structure of the myofilaments within the electron microscopic view of that cell, if you will. It's a phenomena which is unmatched in any other organ, just so that you know. I mean, I have huge passion about this cardiac myocell, which I'm sort of containing my emotions to really convey that, but the fact is that it's there. And it's just like your retinal cells have the ability to help you see and appreciate the colors and the vision, or the hearing cells will, they vibrate within the ears to transmit a certain amount of sound, and noise, and music, and etc., etc. The skin cells will help you feel, or help you, ascertain what the touch is. Similarly, every cell has its own function, but the cardiomyocytes, they have this ability called the sarcomeres, and there are tons and tons of sarcomeres within a cardiomyocyte. And each myocyte, if you will, or cardiac myocyte, will have these actin and filaments, actin, myosin, and titin filaments, if you will, anchored like in this fashion. Now, what happens is the actin, which is the thin filament, and the myosin, which is the thick filament, they slide in this direction and this direction, so that there is a crossover, and these are called cross bridges. Now, the cross bridges, they are being anchored by what you may call titin, and these titin filaments, they are supposed to be the longest and the largest proteins that exist within the human body we have found so far, and they're curled up like this. They're like the springs of a ballpoint pen, if you will, electron microscopically. So the more you stretch the spring, the greater is the tension that will just retract back. So try stretching a spring. it comes right back. Try stretching the spring, it just comes right back. So similarly, these proteins, they could be stretched. You see how I'm showing a stretched protein here? And that's the actin filament, the one in the black. And these actin filaments, they're anchoring right here. Now that actin filament itself has its own rigid part and then the non-rigid part. The non-rigid part is obviously the titin filament, excuse me. The titin filament has its own rigid part and the non-rigid part. The non-rigid part can stretch, and therefore when this myosin filament goes in this direction, this stretches out like this. And that perhaps will lead for greater contractility. The more the stretch, the greater the push will be for the myosin to come back in. And that now we are starting to understand that the Frank-Starling mechanism, which we have learned at our med schools like four decades ago, or for years we have known this Frank-Starling mechanism, or the curve, that longer the stretch, the greater is the contractility of that heart muscle, has now come to an understanding that it is because of this titin filament that stretches out like this because the myosin is moving out, and as it comes back in, the filament just contracts or retracts back. So that perhaps explains that. It'll now shedding more light also that we are starting to understand that many of the cardiomyopathies, for instance, the dilated cardiomyopathies or the hypertrophic cardiomyopathies, may be related to this titin filament. And some of the titin genetic variations and mutations that occur, they are all centering down here to explain some of these cardiomyopathies, if you will. And hopefully we'll have a better understanding about how this works in the next several years. But there's a huge amount of work that's going on, and it's unfolding now into various articles and journals. So as I said, it's also postulated that when, say, the heart muscle hypertrophies in a setting of aortic stenosis or hypertension or idiopathic hypertrophic, for instance, there's a lot of a chaotic type of dyssynergic type of contractility that's occurring within the septal wall, which is hypertrophied asymmetrically. Those patients have, we are thinking now that actin could be responsible for many of that, like the hypertrophy, for instance, the concentric hypertrophy or the asymmetrical hypertrophy. It could be all related to the titin filaments, which either are performing too well, or they're not performing, or they're performing suboptimally, or they are performing normally, depending upon what the situation or the condition then there is. So in a nutshell, the sarcomere is consistent of a unit between these two Z lines, and the two Z lines consist of huge number of actin and myosin filaments, which are anchored from one end to the other. The actin are the thin filaments, myosin are the thick filaments, and obviously they are sliding over each other to make that contractile unit, the sarcomere, function. And millions and millions of sarcomeres across these cells within the myocytes, they constitute the myofiber, help the myofibers contract and relax, so the heart muscle then contracts and relaxes and carries on the pump function. I hope you found this interesting, because this is the mechanics and the entire machinery of the heart at the cellular and subcellular and the microscopic level. And the biochemical changes that occur obviously are related to the calcium pump we talked about in the previous video. And these are actively functioning pumps which require ATP energy. The calcium moves in, invaginates within the sarcoplasmic reticulum, triggers the calcium which is already existing there, and there's a whole chain reaction which leads to this contractile mechanism that we just talked about within the sarcomere between these two Z lines. Okay. Well, I want to thank you for your attention, and keep watching on MuniMeterHealth.com, because we are on YouTube and Facebook, primarily because of the MuniMeter Health, which has contributed a great deal of media for us to communicate and talk to you distantly, far and near. And I wish and I pray that you stay well. And at the same time, I want you to also write back a couple of lines and tell me how the video presentations went. Any suggestions, any comments that you want to make, press the button. And of course, don't forget to subscribe to my MuniMeterHealth.com channel on YouTube. Okay, this is Dr. Jaitly, and see you again in my next presentation very shortly.